We consume millions of pounds of honey, pouring it into tea and bread. Yet the architect of this sweetness is surprisingly small. We see a standard drop of honey break apart. Over its lifespan, one bee produces only a 1/12 of a teaspoon. To understand the scale, compare it to the raw material. On the left, thin, watery flower nectar. On the right, dense honey. The physical gap between these two liquids is enormous. How do these tiny insects actually remove all that water and convert a thin floral juice into a sticky, energy-dense syrup? The answer comes down to a massive, highly coordinated biological assembly line. Every step is an engineered solution to a singular, vital problem, ensuring the colony has enough calories to survive the winter. The process begins out in the field. Foraging worker bees fly from bloom to bloom, drinking the sugary nectar. A single bee might visit hundreds of individual flowers during just one collection trip. They do not carry this liquid externally. Instead, they swallow it, holding the nectar inside a specialized internal organ called the honey stomach, which keeps it completely separate from their normal digestive system. Once a forager returns to the hive, she regurgitates the liquid, transferring it directly from her mandibles into the mouth of an indoor processor bee. During this specific mouth-to-mouth exchange, the bees secrete digestive enzymes into the liquid. These enzymes immediately get to work breaking down the complex sugars in the nectar into simpler, more digestible components, like glucose and fructose. This biological handoff chemically alters the raw material. With the molecular breakdown underway, the colony now has to rely on pure physics to finish the job. The processor bees carry the altered liquid deep into the hive and deposit it directly into the hexagonal wax grid of the honeycomb. At this stage, the substance is still far too watery. If left alone, the high moisture content would cause natural yeasts to ferment, ruining the colony's food supply. To solve this, hundreds of worker bees line up over the open cells and rapidly flap their wings. Together, they generate a continuous, highly targeted draft through the hive. Looking at this cross-section of a honeycomb cell, you can see how the airflow works. As these artificial wind currents pass over the surface of the liquid, they strip away the excess water molecules, accelerating the rate of evaporation. The volume of the liquid steadily drops until it condenses into a thick syrup. Once the moisture level is low enough, the bees build a solid layer of wax over the opening, capping it tight. By mechanically forcing out the water and sealing the cell, the bees create a completely sterile, shelf-stable vault of calories. It is a guaranteed food source that will not spoil, allowing the colony to outlast the freezing winter months when no flowers are blooming. When you look at the entire sequence, the individual foraging, the enzymatic transfer, and the synchronized evaporation, the sheer mathematics of the operation start to come into focus. This chart shows the input required. To produce just 1 pound of honey, a colony must visit over 2 million blossoms. Reaching them means flying 55,000 miles, the equivalent of orbiting the Earth twice, just to fill one jar. This relentless pursuit of nectar reaches far beyond the hive. As the bees travel from bloom to bloom, they are constantly brushing against pollen, inadvertently carrying it to the next plant. That accidental cross-pollination is the mechanism that allows thousands of plant species to reproduce. If this massive foraging network stopped, the agricultural yields for countless fruits and vegetables would simply fail. The sweet syrup sitting in your pantry is the end result of millions of miles of flight and billions of chemical reactions. It is the output of a perfectly tuned biological machine, one that keeps a single colony alive through the winter, while simultaneously maintaining the broad